Today, we're exploring the key ideas from Life Ascending by Nick Lane. This is an independent educational overview, enriched with our own integrative analysis and actionable exercises. Think of it as a guided reflection to spark your own insights, not a replacement for reading the book itself. The story of life on Earth is not a linear march of progress, but a labyrinthine saga of ingenious inventions, unexpected accidents, and relentless adaptation forged in the crucible of planetary change. From the first flicker of self-organization in primordial oceans to the bewildering complexity of the human mind and the ultimate certainty of death, evolution has ceaselessly refined and repurposed its fundamental components. This grand narrative unveils a universe where biological marvels emerge not from divine blueprint, but from the interplay of chemical necessity, environmental pressures, and the ceaseless drive of natural selection. It is a testament to science's profound ability to illuminate the deepest mysteries, revealing a world far more astonishing in its messy, contingent genesis than any static, preordained design. The journey from simple self-replicating molecules to consciousness itself underscores a unifying principle. Life thrives on disequilibrium, exploiting energy gradients and informational pathways to build ever more intricate architectures of existence, each a brilliant, if often imperfect, solution to the challenge of survival. Understanding this ascent means embracing the exquisite logic woven into every cell, every organism, and every ecosystem. A logic born of billions of years of trial and error, a symphony of resilience and transformation that continues to unfold. This exploration dismantles simplistic notions, instead celebrating the elegant, often chaotic, brilliance of life's relentless climb. From primordial chemistry to proto-life, the architectures of existence. The initial spark of life ignited on a turbulent young Earth, a world vastly different from our tranquil blue marble. Imagine a planet shrouded in a thick haze, its surface bombarded by cosmic debris, its oceans colossal and perpetually churned by immense tidal forces. Yet, within this fierce primordial stew, some 3.8 billion years ago, the raw materials for life began to coalesce. Early theories, like the primordial soup, envisioned simple organic molecules accumulating in shallow waters, awaiting spontaneous complexification. However, this view faltered as geological evidence revealed the early atmosphere lacked the precise chemical composition required for such a soup to thrive. Moreover, a thermodynamically flat soup, devoid of energy gradients, would likely degrade rather than build intricate polymers. The true engine for life's genesis likely resided in the unexpected depths of the oceans, deep-sea hydrothermal vents. These subterranean geysers, discovered decades ago, spewed hot, mineral-rich fluids, creating an environment of profound chemical disequilibrium. While early black smokers offered energy from reactions like hydrogen sulfide with oxygen, early Earth's oceans lacked free oxygen. A more compelling scenario emerged with the concept of alkaline hydrothermal vents. Here, seawater seeping through mantle rocks reacted, creating alkaline fluids that then percolated into the ancient acidic oceans. This interaction precipitated intricate, porous mineral structures, natural mineral cells with catalytic iron-sulfur walls. These labyrinthine compartments provided a crucial confluence of conditions: a steady supply of reactive gases, a means of concentrating dilute organic molecules, natural catalysts to speed reactions, and a stable, long-lived environment shielded from surface calamities. Within these mineral cells, fundamental metabolic cycles, like a precursor to the modern Krebs cycle, could have run backwards, synthesizing organic molecules from carbon dioxide and hydrogen. The challenge remained in storing and utilizing this energy. Modern cells employ ATP, an energy currency, while effective, is inflexible for small energy increments. The breakthrough mechanism, known as chemiomosis, allows cells to generate ATP by pumping protons across a membrane, creating an electrochemical gradient, a natural battery. Crucially, the alkaline hydrothermal vents inherently produced such a proton gradient, as their fluids mixed with the acidic ocean. This provided early life with a ready-made chemiomosic reactor, enabling it to harness energy in tiny, manageable packets and, eventually, to generate its own energy beyond the vents. Thus, the earliest life was probably not a free-floating cell, but a complex, self-sustaining chemistry within these rocky labyrinths, achieving surprising sophistication before breaking free. The informational blueprint of life, DNA, was famously unveiled in 1953, its double helix structure immediately suggesting a mechanism for self-replication. This elegant molecule, built from just 4 molecular letters, accurately copies billions of these letters with astonishing fidelity. Yet, perfect replication is not evolution's goal. Subtle errors, known as mutations, introduce variation, the raw material for adaptation. Beyond its structure, the puzzle of how DNA's sequence of letters translated into proteins, the workhorses of the cell, remained. The genetic code, when finally cracked, proved degenerate or redundant, meaning multiple 3-letter codons could specify the same amino acid. Far from being a frozen accident, as once speculated, this redundancy proved to be a highly evolved feature, often buffering against mutations by ensuring errors lead to the same or chemically similar amino acids. This code within the codons links specific codon positions to fundamental chemical properties of amino acids, suggesting a deep, evolved logic rooted in primordial chemistry, possibly within the very vent environments. The origin of this intricate code, and DNA itself, poses a chicken-and-egg dilemma. DNA needs proteins for replication, and proteins need DNA for encoding. The RNA world hypothesis offers a solution: an early era where RNA, a simpler cousin of DNA, acted as both genetic material and catalyst. In the thermal gradients of hydrothermal vents, small RNA fragments could have concentrated and self-replicated, eventually leading to a prototype code and adapter RNAs for amino acid synthesis. The transition from self-replicating RNAs to protein coding systems was also facilitated by the vent's inorganic cells, which provided a protective, concentrated environment, shifting selection from mere replication speed to the overall metabolism of the nascent cell, fostering the evolution of proteins as master catalysts. Even the ultimate emergence of DNA from RNA finds clues in these environments. The deep evolutionary split between bacteria and archaea suggests DNA replication mechanisms might have evolved independently twice. A plausible scenario involves RNA swarms within the mineral cells being converted into more stable DNA molecules, perhaps through retroviral-like mechanisms, providing a robust genetic archive for the nascent cellular entities. With the fundamental architecture of life laid down, a new energy revolution was needed to power increasingly complex forms. Photosynthesis, the process of converting sunlight into chemical energy, fundamentally transformed Earth. Its most profound innovation was oxygenic photosynthesis, the splitting of water to release free oxygen. This most precious waste imaginable not only formed the protective ozone layer and prevented water loss from the atmosphere, but it also enabled complex, multicellular life by providing oxygen for efficient energy release through respiration. The extraordinary complexity of oxygenic photosynthesis involving 2 distinct photosystems points to a convoluted evolutionary path. It likely arose through the duplication and divergence of a primitive photosystem, with one specializing in pushing electrons to make sugars and another in pulling electrons for cellular energy. A crucial accidental link between these systems, perhaps driven by an excess of electrons from environmental sources like manganese, created a continuous flow. The final revolutionary step was the integration of a tiny mineral-like cluster of manganese and calcium atoms into one of the photosystems. This molecular nutcracker inherently possessed the ability to strip electrons from stable water molecules, unleashing the steady flow of electrons that filled the atmosphere with oxygen, permanently altering our planet and setting the stage for all complex life. The vast chasm separating simple bacterial cells, prokaryotes, from all other complex life, eukaryotes, represents the single most significant transition in Earth's history. While bacteria are models of efficient, rapid replication, eukaryotes are immensely larger and more intricate, boasting internal membrane systems, specialized organelles like mitochondria, and a true nucleus housing linear chromosomes. Crucially, eukaryotes can engulf other cells through phagocytosis, a behavior virtually absent in bacteria, and nearly all engage in sexual reproduction. For billions of years, bacteria dominated, but complex, multicellular life, with its magnificent radiations, is a uniquely eukaryotic affair, underpinning even human consciousness. The energetic cost of maintaining a large, complex cell was a major constraint for bacteria. The origin of the eukaryotic cell is a story of dramatic union, not gradual Darwinian accumulation. Genomic evidence shows eukaryotes are chimeras, a blend of archaeal and bacterial genes, suggesting a fateful encounter between an archaean host and a bacterium that became the mitochondrion. This symbiotic event provided the host cell with a ready-made, highly efficient internal energy system, finally lifting the energetic constraint that had kept bacteria small and simple. Phagocytosis itself, a metabolically demanding process, became possible only with the abundant energy supplied by mitochondria. The nucleus, the hallmark of eukaryotic cells, also finds a compelling explanation in this chimeric context. It likely evolved not merely to protect genes, but to manage a chaotic influx of selfish jumping genes, introns. Eukaryotic genes are in pieces, riddled with these non-coding introns that must be removed before proteins can be made. The nucleus, a membrane-bound compartment, evolved to sequester DNA, allowing time for intron removal before RNA was exported. This accidental solution not only solved a genetic crisis, but also turned introns into a boon, enabling novel gene recombination and leading to the massive genomic expansion and astounding morphological complexity that define eukaryotes. Our very existence, therefore, stems from these profound accidents, a rare symbiotic union that overcame an energetic deadlock, and a happenstance solution to the ensuing genetic chaos. Evolution's dynamic toolkit: movement, perception, and replication strategies. With the fundamental cellular architecture in place, evolution began to equip life with an astonishing array of dynamic capabilities, shaping the interactions that define ecosystems and driving the diversification of species. Among these, sex stands out as perhaps life's most peculiar and counterintuitive invention. From a purely objective standpoint, it appears profoundly disadvantageous. It shuffles successful gene combinations, exposes organisms to diseases, and incurs immense costs in finding a partner. Most strikingly, it carries a twofold cost, requiring two parents to produce one offspring, effectively halving each parent's genetic contribution compared to simple cloning. Yet, despite these numerous drawbacks, virtually all eukaryotic organisms engage in sex, while asexual lineages are rare and evolutionary dead ends. This universality signals overwhelming, immediate advantages. Early theories offered partial explanations. Sex generates variation for natural selection, or purges accumulating detrimental mutations that would otherwise doom asexual populations. The Red Queen hypothesis posited sex as a perpetual arms race against rapidly evolving parasites, constantly generating new genetic combinations to stay one step ahead. While compelling, these explanations sometimes struggled to account for sex's immediate individual-level benefits despite its costs. A more encompassing modern synthesis points to selective interference in finite populations. In asexual reproduction, genes on a chromosome are tightly linked. Good mutations can be suppressed by bad genetic backgrounds, and strong selection can purge entire chromosome variants, disastrously reducing genetic diversity. Sex, through recombination, breaks these detrimental linkages, allowing beneficial genes to escape their undesirable neighbors and preserving variability. This direct benefit, even if it means fewer offspring, produces fitter individuals in almost any scenario of genetic variability and strong selection. Speculation on sex's origin points to the genetic chaos following the engulfment of mitochondria by the early eukaryotic cell. The influx of foreign genes and rampant jumping genes caused such genomic instability that sex, through recombination, emerged as the only mechanism to forge viable new chromosomes and rescue the nascent eukaryotic lineage from certain doom. The capacity for movement, or motility, also dramatically reshaped life. The very concepts of predation, escape, foraging, and territory defense are predicated on an organism's ability to navigate its environment. The fossil record clearly shows a gear shift in marine ecosystems after the Permian extinction, with motile organisms proliferating and complex ecosystems coming to dominate. Oceans transitioned from a largely anchored world to one bustling with actively moving animals, fostering diverse new lifestyles and accelerating coevolution, such as the explosion of flowering plants alongside their motile pollinators. At the heart of this transformative innovation lies the mussel, a biological marvel that converts chemical energy into mechanical force. Early insights, from Leeuwenhoek's microscopy to Galvani's animal electricity, paved the way for the mid-20th century unraveling of mussel's molecular mechanics. The revolutionary sliding filament theory proposed that muscle contraction occurs as thin actin filaments slide past thick myosin filaments, powered by tiny swinging myosin crossbridges that bind, pull, and release, using ATP as fuel. The exquisite control of muscle contraction by calcium ions flooding the cell to expose binding sites fine-tunes this powerful system. Remarkably, the evolution of muscle is not a simple linear story. Both skeletal and smooth muscle, though sharing the same fundamental actin-myosin system, evolved independently from a common ancestor. Further research revealed the pervasive nature of actin, forming the cytoskeleton, the internal scaffolding of virtually all complex cells. This dynamic structure, constantly assembling and disassembling, underpins myriad cellular movements, gliding, cytoplasmic streaming, and the intricate dance of chromosomes during cell division. Myosin, too, diversified into a superfamily of motor proteins, acting like cellular trucks, hauling cargo along actin skywires. The roots of these sophisticated motor proteins and cytoskeletal elements extend even into bacteria, where structural kinship suggests ancient origins in simpler conformational changes. The majesty of motility, from its most basic cellular origins to the complex power of a sprinting animal, thus arises from variations on the timeless interaction of actin and myosin, a foundational coral that transformed a simple, static world into an exuberantly diverse one. Perhaps no invention demonstrates the power of natural selection more strikingly than sight. While a rarity among all animal phyla, the groups that possess true eyes utterly dominate animal life. The sudden appearance of sophisticated eyes in the fossil record coincided with the Cambrian explosion, suggesting vision was a pivotal driver, intensifying predator-prey dynamics and accelerating evolution. Skeptics once questioned the utility of half an eye, but the answer lies in adaptation. Deep ocean creatures, for instance, might possess naked retinas, highly sensitive to faint bioluminescence, rather than image-forming eyes, demonstrating that even a partial eye, finely tuned to its niche, can be immensely more effective than a complex, full one. Modern models confirm that a sophisticated eye can evolve from a simple light-sensitive spot in surprisingly short geological timescales through incremental, beneficial steps. The evolution of the lens further showcases nature's ingenious opportunism. While some ancient organisms crafted lenses from mineral crystals, vertebrate lenses, including our own, are composed of crystalline proteins. These are often repurposed housekeeping enzymes from other bodily functions, recruited to the eye through simple regulatory switches. This recurring pattern of co-option underscores evolution's pragmatic, cobbled-together approach, building complexity from existing parts rather than designing from scratch. Despite the diversity in lenses, the core light-sensing molecule, rhodopsin, is remarkably conserved across almost all animal life, pointing to a singular, ancient origin. This was powerfully demonstrated by experiments showing a master gene for eye development, PAC6, could induce eyes to sprout on the legs of a fruit fly when introduced from a mouse, indicating a shared, deep genetic blueprint for eye formation. The earliest progenitor of the animal eye might even trace back to algae, which use rhodopsin in eye spots for photosynthesis, suggesting a primordial photocell that duplicated and diversified, giving rise to the myriad eye types seen today. Finally, the evolution of hot blood, a high metabolic rate, dramatically altered the tempo of life. We, as mammals, often take our constant internal warmth for granted, but it's an energetically extravagant strategy, costing 10 to 30 times more than a cold-blooded existence. The crucial distinction is not simply being warm, but generating consistent internal heat day and night regardless of external conditions. The driving benefit for this immense metabolic drain isn't warmth itself, but stamina. Cold-blooded creatures might match mammals in short bursts, but they quickly exhaust their anaerobic reserves. Hot-blooded animals, however, are built for sustained aerobic activity, for relentless pursuit, escape, or intensive parental care. The aerobic capacity hypothesis posits that selection for this maximal activity inadvertently pulls up the resting metabolic rate, leading to abundant internal heat production as a byproduct. The fossil record offers a window into this evolutionary journey, particularly after the devastating Permian extinction, when Earth's oxygen levels plummeted. In this harsh, oxygen-starved world, ancestors of mammals, like the cynodonts, showed signs of high aerobic capacity. Bony palates for simultaneous breathing and eating, muscular diaphragms, and respiratory turbinates for water conservation, all pointing to adaptations for sustained activity. Later, the ancestors of birds and dinosaurs, the archosaurs, also exhibited traits indicating high activity, including a four-chambered heart for efficient blood pumping and feathers, likely for insulation, suggesting warm-bloodedness. Most strikingly, many dinosaurs possessed a bird-like respiratory system with pervasive air sacs and a highly efficient one-way airflow, allowing continuous gas exchange even in low-oxygen environments. A surprising twist relates to diet. Hot blood in early herbivores might have been driven by the need to efficiently process nitrogen-poor plant diets, which cold-blooded creatures struggle with. Ultimately, the emergence of hot blood, our fast-paced way of life, is a tapestry woven from physiological principles, extreme environmental pressures, and the intimate relationship between diet and metabolism, revealing how historical contingencies sculpt life's astonishing diversity. The mind, the body, and the inevitable end. Refinements and finitude. The human mind, with its profound inner experiences and self-awareness, represents one of evolution's most extraordinary yet enigmatic achievements. The hard problem of consciousness challenges us to explain how mere neural firings generate our vivid personal sensations, those subjective feelings or qualia that seem to defy physical explanation. We distinguish extended consciousness, our human-specific, linguistic, autobiographical narrative, from primary or core consciousness, the more animalistic awareness rooted in emotions and motivations, widely shared across the animal kingdom. While language and memory in extended consciousness are increasingly understood as complex programming, the raw subjective feeling remains the central enigma of core consciousness. Our perception of a unified movie in the head is in fact an intricate illusion. The brain independently processes myriad sensory inputs, sights, sounds, memories, before binding them into a coherent yet utterly reconstructed experience. Neurological conditions like visual agnosia, where objects are seen but not recognized, or Capgras syndrome, where loved ones appear as imposters due to emotional disengagement, dramatically illustrate this constructed nature of reality. Targeted brain stimulation can reliably trigger specific sensations or emotions, reinforcing that consciousness emerges from specialized, collaborating brain anatomies. The brain integrates this fragmented data through dynamic neural synchrony, with different brain regions firing together in coordinated oscillations, strengthening connections and forming a unified perception. Our sense of self, a proto-self, might arise from hierarchical neural maps charting bodily states and how external objects affect them, imbuing perceptions with emotional meaning. The deepest question persists: how do these neural patterns translate into feeling? Primordial emotions like pain and hunger are urgent sensations driving survival, suggesting that feelings are evolved physical signals. While theories involving mind dust or quantum phenomena exist, they lack robust evidence. A more grounded perspective suggests feelings are entailed by specific, reproducible patterns of neural firing shaped by natural selection. The perceived immateriality and ineffability of feelings might even be functional adaptations. To survive, the brain operates on a need-to-know basis, shielding us from direct introspection into its physical mechanisms, fostering a sense of self distinct from our biological machinery. Feelings, as a precise, non-verbal neural language, are then imperfectly translated by verbal language, yet they anchor words and enable shared understanding. Evidence from children with severe brain abnormalities and from animal studies suggests that the roots of consciousness lie in ancient, shared brain regions, implying the firing-to-feeling transformation is less mystical and more widespread, perhaps even extending to creatures as simple as an ant seeking a sugar reward. Ultimately, consciousness, at its core, is a vibrant neural code imbued with meaning by evolution's crucible, rooted in the fundamental experiences of life and death. The human quest for immortality, a timeless aspiration, often overlooks a critical distinction: the quality of endless life versus the inevitability of aging and decline. This fundamental tension is not a cosmic flaw, but an evolutionary design with profound implications for our understanding of disease. Death, far from being accidental, is an ancient invention of life itself, evolving for the benefit of genes, not necessarily the individual organism. Early single-celled organisms, like cyanobacteria, demonstrated programmed cell death, a deliberate self-liquidation to ensure the survival of the genetically identical collective. This sophisticated machinery, involving caspase enzymes, laid the groundwork for complex life. As organisms transitioned to multicellularity, a crucial distinction emerged between the immortal germline, carrying genes to the next generation, and the disposable soma, the body designed to support it. Specialization in multicellular life demanded policing. Cells attempting independent proliferation were punished with death, thus preventing uncontrolled growth like cancer. The mitochondria, themselves ancient bacterial symbionts, bequeathed this death apparatus to eukaryotes, embedding it into the very fabric of complex life. While cellular death is essential, the aging and death of whole organisms require further explanation. Early ideas that aging simply rids populations of worn-out individuals proved circular. A more robust Darwinian perspective highlights a statistical likelihood of death from external factors. Natural selection cannot eliminate genes whose detrimental effects manifest after reproductive age. These late-acting genes accumulate, causing diseases of old age, effectively unmasked as we eliminate early life threats. This antagonistic pleiotropy describes genes beneficial early in life, e.g. promoting reproduction, that carry a hidden cost of later life degeneration. However, a revolutionary discovery challenged this fatalistic view. The identification of gerontogenes in model organisms revealed that single mutations could significantly extend healthy lifespan. This demonstrated that aging is not irrevocably programmed but pliable. The underlying mechanism involves a fundamental evolutionary trade-off: sex or longevity. When resources are abundant, insulin and insulin-related growth factors signal the body to invest in reproduction. When scarce, these pathways quieten, activating genes for bodily maintenance and preservation, effectively putting life on hold to await better reproductive conditions. This explains why calorie restriction consistently prolongs life and health span, often at the cost of fertility. Key molecular hubs like SIRT1 and TOR mediate this switch, offering tantalizing targets for anti-aging therapies. The true orchestrator of aging appears to be the mitochondria, specifically the subtle leak of free radicals during respiration. Though initially conceived as random cellular attackers, free radicals are now understood as vital signaling molecules. While antioxidants often interfere with these signals, a controlled reduction in free radical leak consistently correlates with longer, healthier lives across species. This chronic low-level free radical leak eventually triggers a pro-inflammatory state within cells, altering their internal environment and unmasking the detrimental effects of those late-acting genes. Cells respond in two ways. Some, like neurons, unable to switch to alternative energy sources, die, leading to degenerative diseases. Others, like astrocytes, can adapt and proliferate, contributing to cancer. Calorie restriction works by lowering free radical leak, fortifying mitochondria, and resetting this cellular clock, thus suppressing both cancer and degeneration. A genetic variant in human mitochondrial DNA, for instance, has been linked to significantly reduced age-related hospitalization and increased longevity. The message is clear: aging is a pliable evolutionary variable. While ethical and practical debates surround extreme lifespan extension, evolutionary theory suggests that a panacea against the diseases of old age is not a myth. By targeting the fundamental mechanisms of aging rather than just its symptoms, we can aim to significantly prolong health span, offering a future of vigorous, disease-free golden years. Integrative Analysis and Implications. The expansive journey through life's fundamental inventions reveals a profound tapestry woven from a few recurring threads. Central among these is the relentless interplay of chance and necessity. Life's genesis in hydrothermal vents, the peculiar degeneracy of the genetic code, the accidental integration of a manganese cluster for water splitting and photosynthesis, and the fateful encounter that birthed the eukaryotic cell, all underscore that while chemistry imposes strict rules, necessity, the precise pathways taken often hinge on contingent events, chance. These accidents, once they provide a survival advantage, are then ruthlessly refined by selection, becoming the bedrock for subsequent innovations. Another pervasive theme is modularity and repurposing. Evolution rarely invents entirely new components from scratch. Instead, it ingeniously adapts existing elements for novel functions. Crystalline proteins in our lenses were once housekeeping enzymes. Mitochondrial death machinery, inherited from ancient bacteria, was co-opted for programmed cell death in multicellular organisms. And the fundamental actin-myosin system, initially involved in simple cellular movements, was repurposed into the powerful machinery of muscle. This resourcefulness, a hallmark of biological innovation, demonstrates that complex structures often arise from the clever rearrangement and specialization of simpler pre-existing parts. The concept of trade-offs is equally fundamental. Sex, for all its advantages, carries a heavy cost. Hot blood provides stamina, but demands immense energetic expenditure and often correlates with shorter lifespans. Evolution is a master of compromise, balancing competing demands for survival and reproduction. Similarly, co-evolutionary arms races, like the ongoing battle between hosts and parasites, relentlessly drive adaptation and diversification, ensuring that static solutions are always temporary. Underlying all these mechanisms is the fundamental role of disequilibrium as a driving force. Whether it's the proton gradient in a hydrothermal vent, the energetic difference in a photosystem, or the constant need for new genetic combinations to outwit evolving pathogens, life exploits and generates gradients of energy and information. This ceaseless seeking of equilibrium through dynamic disequilibrium is the engine of biological change. This grand narrative, spanning billions of years and scales from molecular to planetary, underscores the profound explanatory power of evolutionary theory. It offers a coherent framework for understanding phenomena as disparate as the precise sequence of DNA, the structure of an insect's wing, the intricate dance of consciousness, and the inevitability of death. It challenges simplistic, dogmatic views, instead presenting science as a continuous, iterative process of observation, experimentation, and refinement. Our understanding is always provisional, constantly evolving as new evidence emerges. The implications of this journey are far-reaching. By unraveling life's inventions, we gain profound insights into our own biology, from the molecular basis of disease to the evolutionary roots of our minds. The lessons from photosynthesis offer pathways to clean energy, while understanding aging provides tantalizing targets for extending health span. Ultimately, this scientific exploration offers not a diminution of wonder, but an amplification of it. To comprehend life's astonishing ascent with all its contingencies and clever solutions is to glimpse the true majesty of existence, a testament to the enduring human eagerness to know, to question, and to marvel at the intricate, self-organizing brilliance of the living world. Actionable exercises. 1. The vent for new ideas. Inspired by life's origin in turbulent hydrothermal vents, create your own vent for new ideas and personal growth. Choose a specific area where you want new insights or habits to emerge. This vent could be a dedicated journal, a brainstorming session with a diverse group, or a focused hour of solitary reflection. The key is to deliberately introduce disequilibrium, new, challenging inputs, unexpected perspectives, or even discomfort, and allow them to concentrate within this dedicated space. Resist the urge to let ideas dissipate. Instead, let them react, clash, and merge. This focused, contained pressure can catalyze the emergence of truly novel solutions and pathways for evolution in your life. 2. Repurpose your molecular tools. Just as life cleverly repurposed existing molecules to invent sight and harness the sun, look at the molecular tools you already possess. These are your existing skills, experiences, routines, and even past mistakes. Instead of seeking entirely new solutions for challenges, identify one current goal or problem. Now, brainstorm ways to repurpose an unrelated skill or a forgotten lesson from your past to tackle it. Can you combine two seemingly disconnected experiences to create a novel approach? Look for accidental connections between different areas of your life. This exercise fosters creativity and reveals hidden potential, demonstrating that innovation often comes from recombining what's already present. 3. Your inner nucleus for focused evolution. The complex cell developed a nucleus to manage internal chaos, allowing for sophisticated evolution. You can apply this by creating an inner nucleus in your own life to foster focused evolution. Identify a significant goal or project that feels overwhelmed by distractions, conflicting priorities, or fragmented information, your junk genes. Design a specific time and space, your nucleus, where you deliberately sequester yourself from these external demands. During this time, focus solely on processing, organizing, and splicing out the unnecessary, giving your mind the necessary quiet to work through complex problems. This dedicated mental compartment allows your ideas to mature and evolve without external interference, leading to deeper insights and more meaningful outcomes. 4. Practice genetic recombination for adaptability. Life thrives on genetic recombination, the mixing of genes through sex to stay adaptable and overcome challenges. Apply this principle to your own evolution. Identify a routine, a way of thinking, or a problem-solving strategy that has become rigid or less effective. Now, actively seek out ways to introduce recombination. This could mean collaborating with people who have radically different perspectives, deliberately trying an unconventional approach to a familiar task, or exposing yourself to completely new fields of knowledge. The goal is to break the links of ingrained patterns, create novel combinations, and generate the variability needed to adapt, innovate, and thrive in an ever-changing world. 5. Build your aerobic capacity for life's challenges. Just as hot blood evolved to provide stamina and sustained energy for life's challenges, you can build your personal aerobic capacity. Choose one area of your life, intellectual, physical, emotional, or creative, where you want to enhance your endurance and resilience. Instead of just maintaining a comfortable baseline, commit to consistently pushing your maximal activity in that area, even if it's just a small intentional increase each day. For example, if it's learning, dedicate a consistent, focused period to a new challenging topic. Observe how this sustained effort not only improves your performance in that specific area, but also elevates your overall resting metabolic rate, your general energy levels, mental clarity, and readiness to tackle other demands in your life. That wraps up our exploration of today's book. If even one idea here gave you something to reflect on, give it a like so YouTube knows to share more with you. And if you'd like the next insight in your feed, hit subscribe. Remember, what you heard today is our independent interpretation, enriched with analysis and exercises. The full book has far more depth, so consider reading it to experience the author's work directly. Thanks for listening to DeftU Insights, where we turn great books into tools for growth.